Okay.
Okay. Hi, and welcome. I'm Anne from the EMBO Journal. We're doing this little Google Hangout session to give you guys a chance to figure out how we work as editors for the EMBO Journal, to ask questions. Anyone who's watching can write questions to us via the Q&A. They should then appear on the side, and we will try to answer the questions as they come in. Just say I'm joined here today by my colleague Karin.
Hi, and so I'm talking in from Heidelberg, and Anne is in New York. And so we're doing sort of a cross-Atlantic Google Hangout.
Yeah.
I'm an editor with oh, go ahead, Anna.
No, I just want to say, yeah, I'm at the Cold Spring Harbour transcription meeting at the moment. So I'm sitting in my lovely room here at Cold Spring Harbour. So this is also scheduled now today at this time to also give people in the US a chance to talk to us live and send in their questions. Yeah, so the way this works is that Karin and I will be going over some different topics. We'll talk about, as I said, a little bit about how the publishing process works at EMBO, what we as professional editors look for in papers, try to cover some of the questions we're frequently asked at conferences, and then if anyone has comments or additional questions, you're welcome to ask them at any time point.
So both Anna, as Anna said, both Anna and I, we are editors with the EMBO Journal. And the EMBO Journal is a journal that covers really a broad area of molecular biology. The EMBO Journal is published by EMBO Press, and EMBO Press is an umbrella situation that has 4 journals under it. So there's the EMBO Journal, which is where both Anna and I work. There is EMBO Reports, which has a similar scope as the EMBO Journal, so it covers pretty much all areas of molecular biology, but has a sort of a more shorter format. Then there's also Molecular Systems Biology, which is a journal that focuses mostly on systems biology, and then there's EMBO Molecular Medicine, which has more of a translational angle. And so I think with these 4 journals, we're covering a wide scope. And I think if you look at the content that the 4 journals publishing, we're publishing really, really nice papers. And so you should take a look at our website and the papers that we publish, and I think you would be very pleased with what you see.
Yeah, and the way it works, so we are quite a small publishing house. So we have 4 journals, as Karin just mentioned, in-house. And we're a team of around, I think, across the 4 journals, we're 12, 13 professional editors. Everyone came directly from a postdoc. We all have very different scientific backgrounds, which also reflects the categories and areas we handle within the journals. And we really deal with the scientific aspects of the papers, of the analysis. So we deal with manuscripts as they come in, read them through, work with referees. We don't do much in terms of copy editing and marketing. Since we're a small publishing house, we do that in collaboration with Wiley for production and typesetting of manuscripts. One thing I want to say, so I mentioned we're teams of professional editors. At the EMBO Journal, we are currently 6 editors with different nationalities. We all, of course, have English as a work language. We all came, as I said, from a postdoc. In my case, I worked in the RNA field originally, did both my PhD and also partly my postdoc working with RNA-protein interactions, RNA processing, RNA modifying enzymes. And as a result of that, or fitting with that, I'm currently dealing with everything RNA biology for the EMBO Journal, but also the chromatin transcription field, which is why I'm currently at the transcription meeting. I also deal with translation, protein folding, and some structural biology. So as you can see, that's a typical combination of topics, that we have 3 core areas that reflect the areas we used to work in. And since the journal is very broad, we also have people with very diverse, more, yeah, Karin will tell you as well, more developmental background or more trafficking background.
So the way it works, so when you submit a paper, you'll submit a paper online. And so the way the process is that the first thing that happens is that your paper or manuscript will be assigned editor. And this depends on the subject of the area. As Anna mentioned, Anna was RNA, chromatin transcription. I come more from a developmental neurobiology field, and so I cover neuroscience, immunology, as well as some host pathogen. And so depending on the topic, the paper will be assigned to an editor. Then the manuscript will be on my desk or Anna's desk, and then we read the manuscript. We are always asked if we only read the cover letter or the abstract, and I can tell you we read the paper in full. So For me, the cover letter is not so important. For me, it's really the figures and the results section. And I think each editor has their own way of reading a paper. Yeah. So for me, it's very important to look at the figures and the results section. So once a paper has been assigned to an editor, the editor reads the paper in full, the relevant literature, and then we have a very good team spirit, and that's all the 4 EMBO Press journals, that we discuss lots of the papers with each other. So we have a daily 2 o'clock meeting, which is sort of a mini journal club every day, where we discuss papers where we'd like input from other editors on. And these meetings are really, really very helpful, because we discuss the things we like about the manuscripts, but also the things where we're a little bit unsure. And out of these discussions, then we take away from the meeting, well, what's the next step should be.
And I think that's also, just to add to that, I think there's really a strength to also have a very broad background of research areas and expertises in the field, because being, especially for the EMBO Journal, we really strive to be a general interest journal and publish papers that are really of broad interest also beyond the sort of more narrow scope of a single field. And there it's really helpful so when you submit a paper, and one editor, of course, is responsible for reading the manuscript through, but in most cases, additional people are involved in commenting on the story. You get different angles to it and make sure that it's really seen by multiple angles. What we also tend to do with the meetings, and of course, in some cases, we may be a little bit unsure exactly how one places the advance of a given manuscript. We have the option, if we can't, we discuss it with the team, we're not fully sure where to place a certain study. We have an editorial advisory board of active scientists distributed in different fields. And we may then contact them with specific questions, saying this, outlining the manuscript, saying this study shows this and this and that, but we're not fully sure how to place this in the context of the field. We ask them for their additional input, which also makes sure that we stay very much in contact with the active field, where the field is going, where the interests are.
So after an editor has read the paper, or we have discussed it with our teamworks, or we have gotten advice from the advisory editorial board, then we take a decision on the paper. And the initial decision is either to send it out for full review, or to send it back to the authors with an explanation why we think it is not a good fit for the EMBO Journal. And the benefit for the authors is that they do get a fast response from us. And so if we have a paper where we feel that it has certain limitations, so the EMBO Journal is a journal that we like a complete story. So we like to understand for example, what a protein does. We like to understand the mechanism. We like to understand the phenotype. So we're looking for fairly complete papers. And so at times when this also depends on the novelty of the protein or the gene we're looking at. And so if there are cases where we find that we need more molecular insight into how this protein works, we will send it back to authors and say specifically, in this case, we would need some more insight into it. Mm. If there are novelty issues with the manuscript, we will also say, we find that the paper is not a good fit for us because of novelty issues. Yeah. Mm. And so we try, when we give feedback to authors why we think a paper is not a good fit for us, we try to be as specific as is possible.
So one thing that we also often like to tell people and really want people to explore more is that this is, of course, a lot of issues and things that happens at a journal. This is how we work as editors. Papers get submitted, we read them through, we make decisions, we look for referees. But as an author, you often feel that this is a bit of a black box. You send your paper in, you wait to hear back, you don't really know what's happening, you don't really know what the basis for the decision is, you don't know, if you read a paper in a journal, what were the comments raised by the referees, what actually happened that allowed this paper to be published. And what we're doing in that aspect, and sort of for all the EMBO Press journals, to try to make that entire process more transparent, more open, more easily approachable for authors and scientists in general, is that a lot of this information, so we didn't talk so much about the review process, we'll do that a little bit as well later, but both the letters from the editor and the comments from the referees, as well as the response from the authors when a paper is published, we actually publish that alongside the manuscripts that we have. So if you look at our website, for instance, you want to download the PDF of a given manuscript, next to the PDF there's a little button that gives you the review process file, which is a PDF that contains a transcript of, as I said, the communication between the author and the editor, the referee comments, and the response from the authors. And for us, this is a very nice way of trying to keep the process as open to, we can discuss what were the problems of a paper, how were they addressed, see what was raised, and in that way showcase how we as editors actually think about papers and which grounds we use for making our decisions.
So as Anna mentioned, once we have decided to send a paper out for review, the next step is to find the referees, and that's probably one of the most critical. It's a very important part of our job to find the right combination of referees. And so we are looking for experts that can assign, of course, the different technical aspects of the paper, but we're also looking for people that that will give us their opinion on if they find if it's a significant advance or not. And so usually the way it works, we will go to 3 referees. If there are several, sometimes papers have multiple techniques that need to be covered, and then we will have additional referees involved. And we're looking for, what we're looking from referees is really constructive referee reports. We are very selective in the papers that we send out for review, and for the papers we send out for review, we want a constructive feedback from referees. And so we like to use referees that we know from the past will give us constructive comments on the paper. And so we'll go to the 3 referees and get them comments back. And based on these comments, we then make the decision on the paper.
And one thing to add to that, so that is really something that is a very critical aspect of dealing with papers as an editor, because you want the right referees to look at the paper. Of course, we also know that scientists are busy, referees are busy, and in some cases we always strive to make sure that decisions are made in a fair amount of time. Normally we give our referees 10 days to return their reports, but of course there's cases where you really want to make sure you have the right experts look at the paper. So then as an editor you also look, maybe I will give this person a bit of extra time because I really want to make sure I have the right expertise, the right people look at the manuscript. Even so, you often have cases, and especially as Karin just mentioned, sometimes you may have a manuscript that intersects 2 different fields, or a manuscript where you have a strong technical aspect. Maybe you have structural biology, maybe you have a lot of genomics in there, where you need a technical referee who may be a bit more distant from the field. And it's really a key process to make sure you have all these different sides covered. But since they may also be a very different field, you often have cases where you have very divergent recommendations from the referees as well. So maybe one referee comes back saying this is a fantastic study, it should be published more or less directly, it's really important, it's really answering a big question. And another referee comes back saying that this is really very inconclusive, it's not very interesting, the data is not very good. The third referee may be somewhere in the middle. And then as an editor, you want to make the right decision. And it's not simply a question of saying we have 2 positive, 1 negative, or the other way around. It's not a simple majority call. We've read the paper already, and we have the scientific background as well. So then we really look in detail what comments are raised. Are there some things that are very clearly out of the scope of the study? Did someone overlook something? Is it simply that it's interesting to one field, not to another? And what we do with that as well is at that point we actually send the referee reports back to the referees and ask all 3 referees to comment back on the reports to see whether they want to add something, modulate, or comment on their own initial report, if they want to comment on concerns raised by the other referees. And in that way, it allows you to not necessarily find a common ground, but if one referee is a strong outlier, either positive or negative, maybe that person noticed something the others didn't, or maybe that person is wrong. So we ask referees to comment back, and we have a better basis for also making an informed decision on the manuscript.
Yeah, so this cross-refereeing, cross-commenting, it works really well, and most referees actually come back with feedback on it. And sometimes it's as simple as saying, I agree with the other referees, or sometimes it's also, I think, let's say there's one referee who says, well, you need more mechanistic insight, and then the other referee might say, well, maybe more mechanistic insight is a second paper. And so it's sort of a way of balancing the comments against each other.
Yeah.
And that are very important, yeah. So once we have the referee reports in, we make the decision on the paper, and that's either to ask for a revision or to reject the manuscript post-review. And I think we have some things that work really well also in the author's benefits, when we ask for revisions on the papers. And that is that, one, we allow for 1 major round of revision only. And this is actually in the interest of the authors. And so, for example, if I get a revised manuscript in, and a referee will start issues that was not brought up initially, we will say that when we ask for a revision, what has to be addressed is what was raised initially. Of course, if you're doing a revision and there are some questions regarding some of the experiments that was done during the revision, that's a different question. But if a referee on the revised version says, well, I don't think this is interesting anymore, we don't think this is a fair point, yeah? So what we have one major round of revision, and the other big thing we have is that we have sort of this scoping protection. So if your paper is under revision with us, and that there is a related manuscript that comes out, we don't take that related manuscript into consideration. And so I think this, having one round of revision, having a scooping protection, I think that's a benefit for the author. The one additional thing about the one round revision is that we don't have at any of the journals at EMBO Press these sort of endless rounds of revision. So usually we have a very tight process. You usually have one major round of revision that I think on average lasts 3 months. You might have another minor round of revision to tie up some loose ends, but we avoid these long processes where we ask for one revision after the other. And I think this is a benefit for authors.
Which also means, as just to add to that, as an author, because we do this one round revision, and we really want to, we don't want to lead people on and have them do experiments that then are turned down by the referee. So we also normally try to actually define quite clearly what is required for the revision when we invite the manuscript. So as Karin said, we normally say 3 or 4 month time for the revised version. So if referees insist that you would have to do experiments, it would really take you additional 2 years before they would be conclusive, then we don't want to say, well, come back in 3 months, which is impossible, and then you will work for 3 months and the referee will say it's still not good enough. In some cases, if the referees express strong interest in the paper, but they also have a lot of concerns, we sometimes go back to the author at that point saying the referees would require a lot, but how much, what could you imagine adding within 3 or 4 months? The authors will say, well, we can do these points, will this be enough? And then you try to find a ground of saying, well, if these experiments can be done, and if they work, then we will more or less, of course referees will still have to look at it, but you still agree on what is required for the revision. And if it's something that's really very further reaching, would be a very unclear outcome. We also don't want to hold authors to a promise of saying, well, you have to show exactly that, even if it's not clear if it's even happening or not, because then you never know where the data takes you. So we try to really define what is required for the revision, which also means that because we make this very strong decision at that stage, when we invite a revision of a manuscript, there's actually a very, very high rate of those. So I think 95% of the invited revisions are then eventually published because we define very clearly what is required for the revised manuscript.
Yeah, and just to add to that, what Anna said, when we ask for a revision, we make a strong commitment to the paper. It's pretty much, we would like to see the paper published in the EMBO Journal and would like to work with authors on getting it published. Of course, when things go wrong or there are some experiments that doesn't work out, you can't control that for that. But I think, as Anna said, it's about 96% of papers where we ask or invite a revision, they get accepted within a rapid time frame. I think one thing I want to add is that sometimes what I do notice, and I'm sure Anna noticed it too, there's sometimes I have a revised version that's coming in, and I'm looking at the point-by-point response, and I can see that authors really struggle to address one point, and they tried with all sorts of different techniques. And I always wish that they would have contacted me earlier. And so I think what I would like to say is that if you are revising your manuscript and you have questions about specific experiments, feel free to contact the editor. The editor will know how to find the answers for you. And so I think use editors when you have questions and don't sort of be afraid of contacting the editor.
Yeah, and similar, there may be situations where again, a bit what Karin said as well, as the referees say, well, we want you to show that process X happens in a certain way, and they suggest an experiment that may be technically very, very difficult, and you as an author actually have a much better suggestion to what experiment one could do, or you have a good argument why that experiment is actually not really adding much to the study at hand. And again, feel free to write us an email, call us, contact us, instead of sort of banging your head against the wall on something that is not really working and not really adding much to the story.
Yeah, so feel free, and you should use the editor as a dialogue with too about the papers. So once a paper has been accepted, we do some additional things that I think is very important for the process as well. So first of all, we publish or we ask authors to provide source data for the gels, and if you're doing immunofluorescence in cells and you have additional examples, you can provide that as source data too. And I think having the source data is really incredibly valuable instead of seeing gels that are really very, very small and very cropped, to see the whole gel is very useful. The other thing that we do is that we carefully check all figures upon the revision stage, and then really look to see if there is do a quality control of the figures. And if we see something that doesn't fit, we will go back and ask for source data, and we'll look into it more carefully.
And what we see here, this is also as much sort of a general encouragement and sort of recommendation for people when writing papers, that as Karin just said, we do screen quite rigorously for any figure, image aberrations, things that look weird in papers that get accepted. And the vast majority of what we see is simply people beautifying. Maybe there was a bubble in the lane, you know, the classic example, you run a gel, the experiment works, but the gel didn't run very nicely, there was a bubble, there was something else. So you cut out the lane, put the two lanes together, and in principle there's nothing wrong with that as such, as long as you indicate, put a line separator in the middle, that this was actually not, because you're portraying it as a continuous gel when it wasn't. So in these cases, it's very easy to solve by just indicating that there was a lane cut out, putting a separator line, and then as Karin said, send us the original gel. You'll put that, we'll see that as source data on the website, so everyone can see that it's fair enough. You show the relevant and the important part of the data in the manuscript, but there was a proper experiment behind this and supporting this. So it's really something just to keep in mind when you're writing manuscripts. How do you present your data? Because for you, you're sure you ran the gel, you know everything is fine. But as the reader of the paper, if you see things where something is portrayed as being one continuous figure, where it's clearly not, because you can see from the pixel structure, from the bands, that this was actually not two flanking lanes. It's just a way of avoiding any confusion or sort of unclear situations. Same with contrasting in figures. We quite often see that you have a Western blot, you have a Northern blot, something like that, where maybe the signal wasn't too strong, maybe the gel's a little smeary. So people tend to really crank up the contrast. So you get a nice black band on a very, very bright white background, thinking that this is now the perfect example of a gel. But you really lose a lot of information because you lose a lot of resolution. It's actually difficult sometimes also quantitatively to see what was this okay? Was there a lot of background that we can't see now? What was the exact, maybe there was a separate smaller band running a bit higher that is now gone because of the contrast setting. And I think an important point is, of course, you're portraying, or you're sort of showing you're showcasing your data, but it's all based on real experiments, and it's okay for your data and your figure to look like real experiments as well. And I think it makes, also if you're trying to reproduce data, you have using an antibody they used in a different manuscript, and you just can't get the same band pattern. Well, if the only thing you look at is a little sort of stamp-sized little cutout band with very high contrast, it's also really difficult to test whether you're properly reproducing it.
And what sometimes you notice too when you ask for source data, there's some labs you will get the source data back within an hour. So it's clear that they are, sort of storing the source data is a priority in the lab. In other labs, it takes a little bit longer time, which is fine too. But it's sort of what I would like to say is that for PIs, it's really important to have sort of systems in place in the lab for where to store source data. And also for PIs to take a look at the source data instead of just sort of the ready-made figures. And I think this is sort of very important and will become continue to become an important problem.
One thing that we're also doing, and this is sort of in a joint effort with guidelines that are set down by initiatives from actually running from the NIH initially, is in terms of reporting statistics, and again this issue of data reproducibility and how rigorous are you with depositing high throughput sequencing data, resource type, proteomics, but also reporting on the state of your cells, your animal model, and especially the sort of rigorousness of your statistical tests, the validity of the different tests. So for that, we now require, when at the moment we do it for revised manuscripts, we ask that authors submit a filled out checklist where they give all this information in one place. Because again, in lab where you normalize your samples, or you set up your statistics, or you do your tests like everyone else in the lab, or the tradition of the lab where people told you beforehand. But what we really want to also do with that is prompt people to think about, well, instead of saying, well, triplicates calculate a p-value, it's all fine, actually think about what kind of question are you showing with your statistics, and is this really validated with the test you're using? And I think this is, a lot of these things are, again, in terms of how you write your materials and methods, how you present your statistics. There are so many small things that affect how easy it is to reproduce or to copy a certain experiment. And trying to actually collect all that data in one place is our hope that this will actually help make it easier for people to also use the data in our manuscripts and set up new experiments based on that.
Yeah, and be responsible with your figures and your data. Yeah, show what you see under the microscope or on the gels. Don't try to sort of make things very extreme. Yeah, be responsible with your figure. This is what your reputation is all about. Yeah, and so that's really sort of an important point.
Yeah. So to sort of add something a little bit more, again, saying that from the side of the author. So you have been writing a manuscript. You had your paper submitted to a journal. It was reviewed, and then it gets rejected. So what do you do at that point? And sort of just to mention, so at the EMBO Press, all 4 journals, we're actually very happy to also look at referee reports from sort of upstream or other journals. So say you had your paper stuck at another journal, and you get rejected, but for more conceptual grounds, not really for the technical comments. What we can sometimes offer to do, depending on the context, of course, how constructive the comments were, is that we can actually look with an arbitrating 1 or 2 additional referees, instead of necessarily doing a new full round of review. We're happy to look at constructive comments from other journals to see whether we could offer to publish in one of our journals based on those comments, especially in situations where the sort of high stakes, lots of competition, you know, your competitor's paper was just accepted somewhere, and you're really trying to not fall behind in competition. That's something we're very happy to do.
And some things that we hear from authors often, it's probably more the PhD students, is that they spend an awful time on reformatting papers. And, you know, none of us really cares about the format, about the paper, you know, how the reference list is. We care about the science, and so don't waste time when you submit to us to reformat a paper. This is really just not very important at this stage. Yeah, of course, in the end, if it gets accepted, it has to have the right format. Yeah, but don't put time into reformatting a paper. It's really not of any importance at that stage.
Yeah, and adding to that, so what we're also really trying, I mean, it has been actually been a possibility for a long time, but we're really trying to actually tell people that like, it shouldn't be that you spend hours and hours trying to submit a paper. So of course, as Karin said, if you get to the acceptance stage, then you have to have everything formatted correctly and have high resolution figures and everything sort of properly labeled. But for the initial submission, we're perfectly happy if you just have one PDF of everything you have, so your manuscript, your figures, your supplemental data, just send us a PDF, and then if the referees like it, if we decide to go ahead with it, then you can do all the rigorous upload individual figures, do all the different proper formatting. So if you have a study and you want to have our opinion on it, you wish to submit it, then just send us a PDF and we'll read the whole paper, look at the findings you have there, and then take it from there.
Yeah. And I think, as one of us mentioned in the beginning, we are often asked about the cover letter. And I think for some authors, writing a good cover letter is something that takes a lot of time too, and a lot of effort is being put into that. And I think the cover letter is not that important. And it's usually, I think for us, as I said, we read the paper in full, we look at the figures, at the data, and this is what we focus our decision on. We don't focus on the cover letter. So it's actually optional if you want to submit a cover letter or not. It's where a cover letter is of use is to mention excluded referees, and that's sort of where the cover letter is good for. Yeah, but I think besides this, if it's troublesome for you to write it, don't put too much effort into it. It's the paper and the science that matters to us.
Yeah. Again, unless you're, as Karin also just said, like if you have situations, of course, there are some cases. Maybe again, there is a competitive situation. There's a sort of very specific sort of context around your submission that you think it's important to tell the editor. But if we sometimes also see that cover letters mainly restate the same thing as the abstract, or even just contains the actual abstract. In that case, it's not really adding much more, and it's not really, again, as Karin said, it's not the cover letter that makes the difference whether or not we decide to send the paper to reviewers or not. That's reading the full manuscript. So don't keep yourself sort of working 2 weeks through the night just to write the perfect cover letter, because that's not, if your manuscript, I mean, your manuscript writes on your data and on the conclusions in the manuscript, not on how you phrase the cover letter.
We are always asked about excluded referees, if it's okay to exclude referees and what we do about it. And so first of all, it's completely fine to exclude referees. I think to exclude 3 referees is fine. If you start excluding 8 of your colleagues, it becomes more troublesome, because we have to have the paper reviewed. And so if you exclude the whole field, that makes it hard for us to get the paper reviewed. But to exclude 3 or 4 people is no problem. And it's if there's competition or you have people that you would not like to say the paper, just exclude them. It's fine. We fully respect excluded referees. Yeah. As I mentioned earlier, we are very selective in what we send out for review. And the things that we send out for review, it's because we're interested in them. And so there's no point for us to go to one of your competitors as a reviewer. And so if you want to exclude somebody, we fully respect that, and we don't go to exclude referees.
And then just sort of to add to that, because then that's another sort of extended question that we then also often get based on the reviewers, is that of course you're free to suggest referees as well. So how do we deal with that if you suggest referees? And for that, it's a bit more. So I tend to think of the suggested referees as a sort of suggestion list for me or starting point for me to look for. Sometimes if they're good suggestions, we may use them. Sometimes we may not use them. Sometimes I use one of them. The problem sometimes there is also we want to, with excluding referees, we don't want to have any negative bias, obviously. But then there are also cases where people suggest their collaborator or their former boss as a referee, and of course we don't want to have positive bias either. We want to have constructive referees that look sort of with constructive criticism at the manuscript. And for that we really go more with referees based on our knowledge of the field. As I said, maybe I take the suggestions as inspiration for who I might look for when maybe I know someone else in the same area who I actually think would be a much better referee. So it's more that way around. The excluded ones we never ever ask. The suggested ones we may ask or we may not, depending on if we have good experience with them, if we find them to be good referees.
Yeah, so like Anna, I sort of do a similar thing. And so to be honest, I actually completely ignore the suggested referees. And I usually, when I read a paper, I have a fairly good opinion, like Anna, of who I would like to go to. Yeah, and it depends on the experts that I know in the field. And if I ignore the suggested referees, if I happen to pick the same referees, well, to me that is sort of there's no bias, yeah. And so I tend to ignore the suggested referees. And I'm sometimes asked if it helps, or if it doesn't help. To me, it makes no difference at all, yeah. One sort of advice that if you do suggest referees, don't suggest your former PI or your collaborators, because it sort of sends a very bad signal. So if you suggest referees, do suggest the right experts in the field. Yeah. What we also get asked is about sort of blinding the author. So when a referee pick up sort of double blinded. So when a referee picks up to review a paper, that this referee will not see who the authors are. And so this is a question that we've been asked, I think, at every talk we give. And what we decided, or what we have done for, I think, the last half a year, is that we allow sort of the manuscript to be anonymized. Yeah. And so if you wish to have your paper or manuscript reviewed anonymously, it's fine. We accept that, and you have to provide us with an anonymized manuscript. Yeah. I don't think we have had too many of them. Do you, Anna, have any? Have you had? I haven't had one.
No, I've had a few, but I don't think I've had any reviewed yet. But it's fairly rare so far. But they are sort of slowly trickling in. So we started doing it, I think, half a year ago, and we're getting a few. But not too many so far. But let's see how it develops.
Yeah. So the take-up has not been so high, but maybe it's just because people don't know. Yeah. So we haven't really had any experience with it so far. I haven't had any experience with it. Yeah. So we do offer it. If it helps or not, I don't know. I think the review process we have in place at the EMBO journals are really a constructive one to begin with. Yeah. I think for some papers, it's also very, very difficult to, once you read the introduction, often you will know which lab it's coming from. Yeah, so I think it can be very difficult, but it's something we offer and are very willing to try it out to see how that works.
So that's just sort of adding or sort of going on from that. So as Karin said, it's something we often get asked about, and this, for us, the aim here is we want to publish very nice papers, but we also want to really make sure that we offer the services and we follow the wishes of scientists working in the field. So as I said, with double-blind peer review is something that we are trying to see how's the pickup, how does it work, and in general, we're very, very happy to hear any thoughts or suggestions to discuss new ideas, new things we could implement, and to sort of go from that, and also sort of with the context of having this Hangout session here today, based with me being in the US, Karin being in Heidelberg, is also just want to mention that as journal editors, we travel to quite a lot of conferences, and we're very, very happy to be approached at conferences, and in general, send us an email, come talk to us about anything. But at meetings, each editor goes to 6-7 meetings a year, where of course the journals are based in Europe and in Germany. So we tend to be a little bit more present at European meetings, but I think for each editor, we go to US, Canada, I would say at least twice a year. We try to also, and increasing numbers, also be present at meetings in Asia. And so for us, it's really an important balance to, of course, our main function is we work from the office, we read papers that come in, but to really go out, to talk to scientists, to see what is happening, where's the field going, how can we do something to help keep or make the publishing process more enjoyable for everybody involved. So both making it easier for the authors, or making it at least, of course, we are inherently in a position where, as a journal, we have to turn manuscripts down because we can't publish everything. But how do we keep that in a constructive way? And are there things we can improve, do differently? Should we change certain things? And for that, we're always very happy to get input and comments from authors, from referees, from readers of the journals.
And one thing that I wanted to mention again, and Anna mentioned it at the beginning also, we publish these review process files for our accepted papers. And if you look, so these are only for accepted papers, yeah. And so there will be, as Anna said, there's a review process file, which shows the communication between the authors and the editor, the referee comments, the referees remain anonymous, and the point-by-point response. And I think these are really incredibly valuable. First of all, it provides transparency to the system. So what went on and why is a paper published? And I think if you can always go and see what sort of what concerns the referees raised, what was the editor's concern, and so you will see sort of the whole time flow and the dialogue. But besides this, I think also there are wonderful teaching tools, especially for PhD students and postdocs, because it's a very good teaching tool to also see how to write a good point-by-point response. And you will see some point-by-point responses that might be a little bit aggressive, where others are really doing a very wonderful job in sort of in replying in a very diplomatic way to concerns. Yeah, and so I think for anybody who is interested, or if you're doing a journal club on a EMBO Journal or EMBO Press paper, yeah, to go and look at these review process files and see what questions and concerns were raised during the process.
I was just a little looking here. I see we actually have a question that came in, which I didn't see on my question list, but I will just, I don't know if you see it, Karin.
No.
So we had a comment from a viewer now asking, and I think a little bit in line with what we just actually covered in talking about the possibility of double-blind peer review. So sorry if we're now overlapping with the point again. But there was just a question now that exactly the problem that as a referee, maybe they will not look at work from unknown labs with the same seriousness and the same concern as that from famous labs. And the question is then what we do to ensure that refereeing is fair. And so in a way, it's that if this is really a concern, we do offer the double-blinded peer review. So you can, as an author, if you're really worried that your name will make a difference for the referees, then you can submit your manuscript as an anonymous manuscript. But I think what we also just discussed, both of us, with how you find referees is that, and I think for that it really helps also that we go to meetings, we know people in the field, and we know if there's some biases around, and we really try to make sure we don't have negative or positive bias in what the referees say and how the referees comment on the manuscripts.
And so the only issue that, one potential issue that could be with this double-blinded review, and it remains to be seen, yeah, because we haven't had very many actual or real cases, is that as an editor, I'm relying on the referee to tell me if there's a conflict of interest, yeah? There's sometimes I'll ask a referee and I don't know if there's a collaboration between the lab or if there's a conflict of interest. And so I'm relying on this honesty from the referees to tell me, hey, I have a conflict of interest in this case, I should not review it because I know the authors well, yeah? And so one thing one has to sort of consider is that if the authors are blinded, that it's sort of harder to detect, or the harder for the referee to get back to us to say if there's a conflict of interest. But as I said, it remains to be seen because we haven't had so many. It's so new still. Did we cover most of the things then?
I think I'm about to say, I don't see any more questions coming in right now. So I think we've sort of had a pretty broad coverage of our policies now. And as we said, so I think we'll probably sort of round this bit up for now. I think, as I said, we've tried to give you an overview of the way that we work as journal editors. Some of the problems that we try to avoid, or some of the policies we have in place to make the publishing process as smooth as possible, as transparent as possible. As I think we've said and repeated it now, if you have any further questions, any more silly things you want to know, big questions, you have a manuscript that you would like our opinion on, you are curious about how one gets into scientific publishing as a career option, you want to know more about the journals, feel free to shoot us an email or give us a call. So if you go to both for the EMBO Journal, but also for our sister journals, EMBO Reports, EMBO Molecular Medicine, and Molecular Systems Biology, on the website you'll find pictures, phone numbers, names, email addresses for all the editors. So just get hold of us if you're curious about something. We're happy with any level of questions and engagement.
Sounds good. I have nothing more to add from here.
Then I think we will round off this transatlantic transmission. Thanks for joining me, Karin. Good night to Germany, and I'll go out and enjoy the sunshine of Cold Spring Harbour.
Okay, bye.
Bye.